Morning, everybody. Thanks for coming. It's my great pleasure to introduce Dr. Myra Delmar. He's a leader of cardiac electrophysiology and arrhythmia research. Dr. Delmar is a professor of medicine and holds the prestigious Robert Martisen Professorship of Cardiology at NYU. His research spans the wide range of topics, including arrhythmia, sodium channel, plaque filling, desmosome, gap junction, connexin 43. Dr. Delmar earned his MS, MD, and a Ph.D. degrees in Mexico. And after that, he completed his postdoctoral training at SUNY Upstate Medical Center in Syracuse. He began his academic career at the Department of Pharmacology at the SUNY Medical School, where he quickly rose to the rank of professor and vice chair of the department. In 2007, he was appointed to the Frank Wilson Professor of Cardiovascular Medicine at the University of Michigan, where he also served as a director of basic science and a co-director of the Center for, the Center for Arrhythmia Research. In 2010, Dr. Myra Delmar joined NYU as a professor of medicine. Dr. Delmar has received numerous prestigious honors, including R35 Outstanding Investigator Award from NIH. His exceptional research is showcased in 132 original research papers and 33 review articles published in high-impact journals, including Circulation Research, Cardiovascular Research, and Nature Communications. And he has contributed 30 book chapters and authored 2 books and delivered 238 invited talks. And his research has been continuously funded by NIH and AHA, and also Reduc as well. And beyond his groundbreaking research, Dr. Delmar has made significant contribution to academic service. He has chaired and co-chaired 8 NIH study sections, including MPPB from 2021 to 23. And he currently served as associate the editor of AJP and the Heart Rhythm Journal. He is also the editorial board of Circulation Research and Heart Rhythm. Dr. Delmar is not only an exceptional scientist, but also a dedicated mentor. He has trained 23 graduate students, including myself, and 34 postdoctoral fellows. I'm extremely grateful to have been one of his students. There is much more things I can talk about Dr. Delmar, but then I will stop here, and please welcome me in joining Dr. Delmar.
Thank you. Thank you, Junko. It's, of all of that list, a lot of it is because I am getting older, so you accumulate more things as you go in your career. But certainly one of the proudest of all of that list is having had outstanding graduate students and Junko being at the top of that. So it is really a pleasure to be here. It's an honor. And of course, Rob and I have been friends since we were very young, and I don't want to say how many years have passed since we were very young, but it is great to see old friends, not because they are old, but because we have been friends for a long time, and to make new friends. And it's wonderful to see these facilities here. It's really an outstanding experience. So what I will tell you about today is about plakophilin 2 arrhythmogenic cardiomyopathy. I modified the title a little bit because I want to talk to you about the molecular mechanisms of the disease. But I also want to bring you into the latest, which is the promise of gene therapy for patients with PKP2 arrhythmogenic cardiomyopathy. So these are the types of cases that we work on. This is the heart of a 16-year-old, 17-year-old boy. He's perfectly normal. He's running around. He's playing soccer with his friends. All of a sudden, this kid collapses. He's in syncope. His heart is not beating anymore. Efforts to resuscitate him are unsuccessful. So a few minutes after this kid has been playing around as a normal teenager, he is declared dead. This is the MRI post-mortem. And what you can see is that this is the septum, this is the free wall of the left ventricle, and the free wall of the right ventricle has lost its muscular appearance. In fact, it has been replaced by the histological section here, you can see it, by adipose tissue and collagen with very little muscle left. The left, on the other hand, it has enough muscle, although there are some collagen infiltrates. So that is why this kid is functioning normally. He can run around because his left ventricle is working okay, but he has completely lost the right ventricle. Importantly, though, this kid does not die because of a mechanical dysfunction of the heart. He dies because of a very severe arrhythmia, and one that did not show any preamble, that was not detectable during his lifetime. This kid is then diagnosed with arrhythmogenic right ventricular cardiomyopathy. This is an inheritable heart muscle disease that may result in arrhythmia, heart failure, and sudden cardiac death, because eventually the disease can progress into biventricular failure. About 50% of the cases of familial ARVC are linked to pathogenic variants in the gene coding for plakophilin-2. And plakophilin-2 is a component of the desmosome. So the best example of a desmosome is in the skin. The desmosomes are making adhesion complexes between cells, and inside they are connected to intermediate filaments. In the skin is keratin. If I pull my skin, the cells don't rip away from each other. And they don't because they are held together by desmosomes that are adhering to the cells. And when I let the skin go, the skin comes back to its initial position because the intermediate filaments stretch. And then when you release them, come back to their initial position. So the intermediate filament in the heart is desmin, but the mechanics are the same. And all of this is a structure that is present at the cardiac intercalated disc, at the point where the cells meet with each other. Some years ago, we developed a mouse model of plakophilin-2 deficiency to study the biology of a protein that in the heart, when it is mutated or absent, causes a disease. So the model is really not an attempt at recapitulating the disease, in the sense that, as you will see when I describe the model, there are many differences between the heart of a patient with a mutation in one allele and taking a mouse and knocking out a gene when they are 3 or 4 months old, completely on both alleles. But it does show us the things that happen if plakophilin-2 is not there, and that has allowed us to understand better the origins of the disease. So this mice model is cardiomyocyte specific and tamoxifen induced. So when the mouse is about 3 or 4 months of age, which is a young adult, is the age at which patients develop or manifest ARVC, we inject them with tamoxifen, and only in the cardiac myocytes we're going to lose the expression of plakophilin-2. We have done it both in male and female, and in both of them, you get about the same result. There is no difference. We monitor the mice for heart function and for survival by means of echocardiograms and electrocardiograms by isoproteironol bolus. And then after euthanasia, we study the fibrosis extent and we characterize the molecular phenotypes. So what happens in these mice? 14 days after we inject them with tamoxifen, nothing has happened. You can see that the walls are okay. They are all muscle, both the free wall of the right and the free wall of the left. This panel is an exaggeration of the blue contrast on the right, just to show you that there is no collagen infiltrates. But if we wait just 7 days more, then we get a different picture. Now, we can see a lot of collagen infiltrates in the free wall of the right ventricle, thinning of the wall, whereas the left is still pretty much in good shape. So what we are having here is a cardiomyopathy of right ventricular predominance at this specific point in the evolution of the disease. And just for comparison, not because I want to tell you that we recapitulate, none of us has recapitulated the disease, neither in mice nor in iPSLs. There are many different features of the disease, but there are some that are consistent, like it is the cardiomyopathy of right ventricular predominance at this particular point in time. Are they, if we follow then this by echocardiogram, you will see 2 variables are important in this context. One is the RV area, which tells us how dilated the right ventricle is. And the other one is the left ventricular ejection fraction. And you can see that 0 is the time of tamoxifen injection. So these time points are days after the plakophilin 2 was knocked out. And you can see that the RV starts to dilate a little bit by day 14, but not really much. But then it progressively continues to dilate. Ejection fraction starts to drop, but really shows signs of a failure by day 28, and then continues to go down. So by this point, the animals are in a biventricular heart failure. And if we follow a Kaplan-Meier curve, what you see is indeed day 21, all of the mice are alive. Day 28, all of the mice are alive, but then the mice start to die. And by day 50, we have 100% lethality. And we have repeated these curves, and we have repeated this time course, and it's quite reproducible. It basically, the model works very well in being reproducible from day to day to day on the evolution of the disease. We have studied primarily these 3 points, 14, 21, and 28. Most of what I will tell you about today happens at this day, which is the state of cardiomyopathy of right ventricular predominance. Are they arrhythmogenic? Yes, they are. If you treat them with isoproterenol, then you start to see arrhythmias. In some cases, you see polymorphic VT. In a few cases, they progress to ventricular fibrillation and sudden death. So we do have a model of an arrhythmogenic cardiomyopathy of right ventricular predominance. In this paper, which is the description of the mouse in 2017, we also did quite a bit of transcriptomics, and we tried to figure out what was the origin of the arrhythmias. And so basically, I just used the title page first to acknowledge all of the people that worked in this project. We are very fortunate to have a lot of collaborations, and so we always are happy to include everybody in the paper. This paper has collaborations from Natalia's lab, from Mike Ackerman in a little bit, a lot from Hector Valdivia, Toon Van Ven. And basically, we came to the conclusion that plakophilin 2 is required for the transcription of genes that control calcium cycling. In other words, there are 2 pieces in here that will be important for the next slides. One is that loss of plakophilin 2 causes a dysregulation of intracellular calcium. And the other one is that plakophilin 2 out there in the intercalated disc being absent causes a change in the transcriptional program of the cell. About 10, now 11 years ago, colleagues at the Johns Hopkins Center had noticed that ARVC patients, plakophilin 2 ARVC patients, did worse if they exercised. And so they systematically studied this, and they noticed, and they basically generated this data. So they took the survival from first sustained VTVF. So how old are the patients when they get their first serious arrhythmogenic event? And they noticed that the athletes, the ones in the green line, are younger when they have their first arrhythmic event. And their hearts actually do worse. The athlete hearts actually do worse than the control. So plakophilin 2 arrhythmogenic cardiomyopathy is one exceptional case in which exercise, endurance exercise, is bad for the patients. Sir?
So given the age range, is that because are there genetic variants, are there SNVs that have different severities that you could map, or is there something else going on?
Yes and yes. So the penetrance of the disease is about 50%. And you can have families in which you have 2 members that have the same variant, and one will develop the disease and the other one will not. Exercise is a major variable in that aspect, but indeed you can have, and the risk, yes. But indeed, the progress of the disease is not linear. Some patients may have a very heavy arrhythmogenic event. What is pretty consistent is that the arrhythmia precedes the mechanical dysfunction. And that is really the most dangerous part. But yeah, the penetrance is low, and there are certainly environmental factors that take place. So why is this? We decided to investigate it, and I am just going to give you a summary of it, so that I can also move to other aspects. And the way we decided to study this was by taking mice, these mice that I have just described, and put them to run on a treadmill. And these mice run for 1 hour a day, 6 days a week, for 6 weeks. And this is what we did it for this particular paper. Now we do 5 days a week for 75 minutes a day, but basically it is the same result, except the postdoc doesn't have to come Saturday. So we changed the protocol a little bit with the same outcome. So this is the paper where we described our results. And basically what happens is that when we exercise, we develop a reduction in the expression of proteins that localize to the intercalated disc. So we found that under normal circumstances, the control mice, when they exercise, they actually have a downregulation of the transcriptome of the intercalated disc. So if you add to that having a deficiency in plakophilin-2, that combined brings you down below the level of tolerance, and you develop a phenotype. We also found that the changes that happen at the transcriptomic and at the phosphoproteomic level significantly affect intracellular calcium dynamics. And so a major component of the arrhythmias that happen in hearts deficient in plakophilin-2, we proposed, comes from a dysregulation of the calcium cycling. And to give that in a diagrammatic way so that I don't bore you with data details, I just use this diagram that I am sure many of you have seen, basically to give you a little bit of a reminder of the calcium cycling. During the action potential in a heart cell, calcium comes in through the calcium channel, meets the cytoplasmic side of the ryanodine receptor. That will make the ryanodine receptor open. The ryanodine receptor is in a specialized component of the sarcoplasmic reticulum called the junctional sarcoplasmic reticulum, where we store calcium. When the channel is opened in response to the entry of calcium, calcium is released. This calcium that is released then goes to the sarcomeres and facilitates the coupling of the actin and myosin to allow contraction. This calcium needs to be then retrieved via 2 ways. One, back to the sarcoplasmic reticulum through the Cerca pump, the sodium, the Cerca pump that is regulated by a small protein called phospholamban. And it can also, if it is too much of it, be extruded via the sodium-calcium exchanger out to the cell, out to the extracellular space. What we found is that there are a couple of things that go wrong in these plakophilin-2 deficient mice and that are exaggerated by exercise. One is a major change in the phosphorylation state of the ryanodine receptor that makes it more eager to release calcium. So the ryanodine receptor doesn't have enough of a break, and during diastole keeps spewing calcium. And the other one is a change in the phosphorylation state of phospholamban. And this is important because phospholamban is the brake for Cerca pump. So if it is hyperphosphorylated, it allows the Cerca to retrieve too much calcium, which fills the cisternae of the sarcoplasmic reticulum. So that when the next action potential comes, there is a lot of calcium that comes out. That excess of calcium out and some changes in phosphorylation here that I will not detail, this excess calcium reflects into an increased activity of the sodium-calcium exchanger. If the sodium-calcium exchanger works extra time, it's electrogenic. There are 3 sodiums that come in for 1 calcium that comes out, which means there is 1 positive charge extra coming into the cell. This is a depolarizing charge, which means if you make the sodium-calcium exchanger work extra time, you start to bring too many sodiums in, and that depolarizes the cell. And as the cell depolarizes, it gets close to the threshold for firing an action potential. So as a result of all of this, what you had is a very arrhythmogenic event in which the more action potentials you get, and with exercise you get more action potentials, you get a faster heart rate, the more you see these changes in the phosphorylation state of the cells, the more you have an overwork of the sodium-calcium exchanger, and then you have this extra depolarization that facilitates spontaneous firings that are happening in the ventricle and that can be very arrhythmogenic. So bottom line, we believe that controlling the ryanodine receptor, we concluded at that time, would be important for that. Those of you familiar with these diseases may see some similarities with what happens in CPVT, right, which is a disease, a very arrhythmogenic disease caused by mutations in RYR2. Important thing is we come to the conclusion that the arrhythmia mechanism is triggered activity, that there is an excess of calcium release from the junctional sarcoplasmic reticulum via RYR2, and this coincides interestingly with observations from Jörn Nollmann and his group, where he published that flecainide, which those of you that may know is commonly seen as a blocker of the sodium channel, flecainide in small doses actually blocks the ryanodine receptor. And it is now the drug of choice for treating patients with CPVT. So we decided to try in the mice what would happen if we give them flecainide, what would happen to the arrhythmias. And the conclusion of that was that flecainide in the mice abolished the ventricular arrhythmias in the plakophilin-2 deficient mice. So the mice, at least, is treatable with flecainide. Good thing is flecainide is an approved drug. So it was just a matter of repurposing it without having to go through all the motions of a clinical trial. But we did go to the motions of a clinical trial. NIH was generous enough to fund this R34 to Hugh Calkins and Wojciech Zareba. So I am not a clinical trialist. I was not involved in it except as a cheerleader, I guess, hoping that this would work. But the results are very interesting. Indeed, they gave flecainide to patients with ARVC that were having arrhythmias. And in fact, the number of ventricular premature beats decreased in a significant manner. The median of those beats decreased in a significant manner. And the number of VTs went down to 0 during the time of the trial. And just to give you a sense of what the arrhythmia burden is for these patients, we're talking with the placebo patients that have about 5,000 beats as a mean a day, 5,000 extra beats in a day. And if you take it as the median, then it's a bit lower, but the range is very broad. So these patients are constantly having these arrhythmias.
How is this drug given to the patient? Where's the half-life of this drug? I mean, it's just the patient needs to have it all the time, or
so they take one pill every 12 hours, I think.
So they need to have it all the time, person.
Yeah.
They need to have it. And okay, so
it's a standard. Those of you that may know the history of antiarrhythmics, for a while it was like the no to go to drug because there was some suggestion that it actually was not beneficial. But now it is being used for example, for patients with AF, and now both in CPVT, and now it is becoming a standard of care in arrhythmia for patients with ARVC. So I showed you this. This is where we started, really, when we saw that there is this change in the transcriptional program of the cell. And we have more recently decided to investigate why is that transcriptional program is changing. So we agree that desmosome disruption alters transcription. I think all of us that are working in this field would completely agree on this. But the question that was bothering me at that time, continues to, is this is an event happening out there in the intercalated disc. So how does the nucleus know? How does the nucleus know that there is a desmosomal complex in the intercalated disc that is getting all messed up? How is that transduced to the inside of the cell? And what I will show you is work that we did in collaboration with Alicia Lundby and Henning Bungard at University of Copenhagen, and was recently, well, not so recently, but was recently published. And basically Henning and his colleagues, and this was really the start point of this project, was able to convince 5 patients, all of them with a pathogenic variant of ARVC, all of them with a diagnosis of ARVC, all of them with ejection fraction that was above 45%, so they are not in heart failure, to subject themselves to an endomyocardial biopsy. Now, that is not so difficult because these patients have a cardiomyopathy. What is really difficult is that they were able to convince 3 healthy relatives to subject themselves to the biopsy so that we could do a comparative study. And so with these extremely valuable samples, we did the transcriptomics, the differential transcriptomics, the differential proteomics, and the transmission electron microscopy. And I will not take you through the whole story. We'll only show you what happened in the EM, because it was very revealing on this, how does the nucleus know? So what I'm showing you here is an EM image of the nucleus of an adult cardiac myocyte from one of the healthy relatives. And if you look at the nuclear envelope is very nice and continuous, is very well preserved through the structure. Some invaginations, but on the other hand, is nice and continuous line. When we looked at the patients, the picture was very different. In this case, what you have are these nuclei that are ruffled with all of these invaginations in the nuclear envelope, changes in the shape and size of the nucleus as well. We were very interested in this because it certainly looked different, and we were curious to know how is that happening. So we first went back to our mouse model and looked in the nucleus of the mice, and indeed, this is a staining with lamin, which is a protein of the nuclear envelope. In the control, the nuclei have a very nice shape. These are nuclei inside the myocytes. We did not dissociate, we do not extract the nuclei. And when you look at the knockouts, you see these are stacks of confocal. You can see that as you flatten this into 2D, you see the equivalent of all of this ruffling that I have described to you in the EM. And indeed, when we compare the intensities of the lamin B, which get more intense because there is more foldings, we see clearly a difference between the two groups. So how could this happen? We took a page from a beautiful paper by Ben Prosser and his colleagues. He studied the interactions between the cytoskeleton and the nuclear architecture. And he came to the conclusion that the shape and integrity of the nuclear envelope is maintained by 2 opposing forces. There are the intermediate filaments, desmin, that anchor and pull away. And then there are the growing microtubules that, as they grow, they push into the nuclear envelope. And there is a balance between the push by the microtubules and the pull by the desmin intermediate filaments. So what he did in that paper was to study the desmin knockdown mice. And what he found was that the nuclear envelope was ruffled, just very much like the one that I just showed you for the ARVC patient. And that is because he did not have the pull force. All that was left was the push force of the microtubule. So we have been interested in seeing whether that could have some comparisons, but in a lot of what I'm going to show you now is using expansion microscopy, because we needed, in order to really study this well, we needed better optical resolution. And so we have developed a methodology for expansion microscopy. And this is one example. This is 100 microns. This calibration is not wrong. This myocyte, single isolated myocyte, measures over 1 millimeter in length, as it has been expanded by a factor of about 8. It has been stained in this case for NAV 1.5, which of course is mostly to see where the Z discs are. And you can see these Z discs perfectly nicely aligned. So the structure of the myocyte is maintained, but by enlarging it by a factor of 8, your optical resolution gains by a factor of 8. We have combined that with structured illumination. And so when you combine both techniques, you gain optical resolution into the 20 to 40 nanometers range. That means when there are 2 points that are away from each other by 40 nanometers, you see them as 2 different points and not as the same point. So this is reconstruction of the 3D image of the nucleus of a cardiac ventricular myocyte. And by the way, all of this is done by a very talented postdoc in my lab, Giorgia Bertoli. And this is a single plane. And the reason why I want to show you this is because we have, as others have found, that the chromatin architecture inside the nuclei, of course, is very preserved and is not just all over the nucleus. If you take a cell in culture, even iPS myocytes, and you stain the chromatin, you just see the chromatin distributed all around the nucleus. In a highly specialized cell like the myocyte, chromatin has a very defined architecture. You have a lot of heterochromatin that goes right under the lamin, and that is called the lamin-associated domain. You see these shells of chromatin that are actually wrapping the nucleoli. And then you see speckled euchromatin inside the nuclei. We have some very interesting results with what happens in the nucleoli of PKP2 deficient myocytes. But I will not talk about that for reasons of time, but I will talk about the lamin-associated domain. So in this hypothesis of Prosser, we want to know what happens to the interaction between desmin and the nuclear envelope. And so using this technique, and by the way, this technique also allows for large areas and three-dimensional reconstructions. So in the next image, series of images, I am showing you desmin in gold, and it's going to be lamin B in blue. And this is, imagine it, that you're going to be moving through the different planes. So we are at the bottom of the nuclei at this point. And as we move, you see that the desmin is reaching the nuclei in a perfectly perpendicular angle in an orthogonal direction with the membrane of the nucleus. So the membrane of the nucleus, the nuclear envelope, is nicely preserved, and desmin has this very nice alignment with the occasional intermediate filaments that go between sarcomere and the other, but for the most part, standing on the Z disc, and when the Z disc gets to the nuclei, it anchors there, like in the image that Prosser developed. So this is the Z stack of all of those images. Again, a few intermediate filaments going between Z discs, but for the most part, this beautiful architectural preservation of the interaction between the intermediate filament cytoskeleton and the nucleus. And that is a single plane, just to emphasize that. When we did the same on plakophilin-2 deficient cells, we got a very different picture. You already get a sense here, even at the bottom of this nucleus, you can see that the direction of the filaments are not really nice and perpendicular to the direction of the nuclear envelope, when we go through the planes, you see, first of all, that the nuclear shape is different. You're going to see a piece of nuclear coming from here that now is showing you that this has like a kidney shape. And the cytoskeleton is not attacking the membrane in a perpendicular direction. In many cases, it's actually running parallel to the nuclear envelope. So this misdirection of the angle of reach and separation from the nuclear envelope is not allowing for maintaining that force that is necessary for the control of the architecture of the nucleus. And this is the stack, just to emphasize the contrast with what I showed you in the normal one. And in the single plane, you see, for example, all of these filaments going completely parallel to the nuclear envelope instead of perpendicular to it. And just to give you a sense of comparison between the control and the knockout, both in the stack and in the single plane. Using more conventional 2D storm, we measured the distance between desmin and the nucleus. And indeed, we observed that the desmin filament separates from the nuclear envelope. So we don't have that force. Now, one of the great advantages that one has in having models of disease in mouse is that one can study drugs without the obvious impediment of giving them to patients. We needed to define what a drug is, and we needed to abide by a strict definition of what a drug is that we're going to use in mouse. So we went by the definition provided by Otto Löw. Otto Löw, as you probably know, is a German pharmacologist. In 1929, he described acetylcholine through a very interesting story that goes outside of the topic, but he dreamt the perfect experiment, jotted it out, went back to sleep, woke up very excited the following morning to realize that he could not read his handwriting. So fortunately, he dreamt it again a few nights later, and that he got out of bed, went to the lab, did the experiment, got the Nobel Prize for it. So if you dream an experiment, don't just write it down. So he was asked, he ended up, because of the war, he went then to NYU, actually. He finished his career there. And he was asked, what's a drug? He thought for a moment and he said, a drug is a substance which, if injected into a rabbit, produces a paper. So we needed to finish the paper. And on the suggestion of the reviewer, we took a drug, and the drug is colchicine. Colchicine prevents the growth of microtubules, and so prevents the microtubule repolymerization. So the thought was, if I give colchicine to a nucleus that looks like this, that would look like this, if I prevent the microtubules from pushing, I may actually preserve the structure of the nucleus. And that is indeed what happened. We did the experiment, and indeed we have a preservation of the nuclear lamina in the knockout if we treat it with colchicine versus the control. Now, the important thing here is that the lamin is protecting the lamin-associated domain. And this is DNA. I'm sorry, you always do this. The lamin is protecting this heterochromatin. And the belief is that cells put under the lamin in a tightly packed chromatin the genomic components that the cell does not need to maintain its identity. So a myocyte does not need the genome of a macrophage. That goes to the lamin-associated domain. That lamin has to be, occasionally, it may open for reading, but for the most part is closed and is not available for reading. We found, though, in an earlier experiment that there is DNA damage in this myocyte. So there is a double-stranded DNA damage detectable. You always detect it in cells, but in the knockout, it was particularly present. And this is increased in exercise. So we decided to look, using the techniques that I just described, at the DNA damage in this nuclei, using the expanded microscopy and the SIM square. And in control, you always found a little bit of DNA damage. In the knockout, though, you find it very close to the lamina-associated domain. That is, DNA that should not be open for reading has opened, and it may be available for reading, and is not myocyte DNA. Quantitatively, we see that there is, the distance to the nuclear envelope is shorter in cells from PKP2 deficient mice than in the control, where the distances distribute more homogeneously. And just in a bar graph, to show you that about a third of the aggregates of gamma H2AX, which is what we mark as a DNA damage repair response, is much more abundant in the knockout than in the control, where the DNA damage is found pretty much homogeneously around. Now, what is happening in these extra openings of genomic DNA that should not be read? That coincided with studies that the lab of Jeff Safitz has done, saying that there is an immune response and an inflammatory response in these hearts that is coming from the myocytes themselves. And that was put into context of this experiment that we did some years ago using the ribotag mouse. And the ribotag mouse is simply a model to be able to pull down what is the RNA that is being read in the ribosome at a particular snapshot. We combined that with our knockout, and to our surprise, what we found is that in cells that are deficient in plakophilin-2, you get a lot of transcripts that have to do with the immune system and with the inflammatory response. So it looks as though there is a loss of cell identity, so to speak, of the myocyte that starts to express genes that should not be expressing and that belong to the inflammatory and the immune response. And this, of course, creates a major complication, because now you can have macrophages coming into the picture and accelerating the progression of the disease. So here I am going to take a little detour to talk to you about what is now happening in terms of gene therapy, because there is a lot of excitement about it, particularly in the area of PKP2 arrhythmogenic cardiomyopathy. So about 3 years ago, there was this rush of interest from industry and from academia to try to use plakophilin-2 arrhythmogenic cardiomyopathy as a model for trying gene therapy in humans. And the reasons are PKP2 ACM is a loss of function disease. So us and others have come to the conclusion that there is no dominant negative mutation. All of the mutations are autosomal, but they are loss of function. The second one is that the gene is small enough to fit into an AAV, and that is, of course, very important. The next is that we cannot overdose with PKP2. So we put PKP2 in a mouse and nothing happens. You cannot overdose it. It tolerates perfectly well the extra. Now, us in cardiology have been very frustrated for many years because cardiomyocytes do not divide. In this particular case, that is actually a good thing, because once a cell takes the gene, it will continue expressing the gene. And finally, for the purpose of industry, PKP2 is a rare disease, but is not so rare that there would be no financial interest on their part for getting into the game. So with all of that, there were 3 companies that came into the fray independently: Lexeo, Rocket Pharma, and Tenaya. We licensed our mice to Tenaya. We did all of the preclinical to Rocket Pharma, and Lexeo did an independent study. And interestingly, we all started at about the same time, and we all published at about the same time, which is between December of 2023 and February of 2024. There were these 3 papers, plus one that came from the Netherlands, from the group of Eva van Rijk, with very similar results. And the important thing I want to say is that all 4 independent studies came to the conclusion that PKP2 gene therapy can be safe for mice and can be effective for mice. So the data that I will show you is mainly from the study that we did, because it was the one that happened in my lab. Importantly, as a disclosure, I had no financial interest in this, either in the past, in the present, or in the future. It was all done as a contract between NYU and Rocket Pharma. So when you take an image of plakophilin-2 in a normal heart, you see this beautiful staining of the intercalated disc. That's the point where the cells come in contact with each other. If you look at the knockout injected only with buffer, you do not see the plakophilin-2 because it has been knocked out. And if you look at what happens after the injection of an AAV that is going to express PKP2, you see the recovery of the signal localizing properly to the intercalated disc, clearly not in 100% of the cells. About 50–60% of the cells take the message. But that is enough to turn a Kaplan-Meier curve with 100% lethality at 50 days to a Kaplan-Meier curve of survival up to 150 days, which is the point at which we interrupted the experiment. So we went from 100% lethality to 100% survival. This is a bit complicated, but it's important to point out, by the way, that the injection here happened at day 7 or 14. We did 2 separate series after tamoxifen. So once we have initiated the disease. And when we measure at the RV area, in the knockout, the RV area is enlarged at day 28. But in those mice where we injected the AAV at day 7, the RV area is preserved all the way to the 5-month point. Those mice that we injected on day 14, the RV area was still preserved, and that continued all the way to 5 months. Ejection fraction showed a similar picture. If you look at the ejection fraction in the knockout, of course, after 28 days, it's very reduced. But when we injected the tamoxifen, we did not go back, we did not go down. Of course, we cannot go up because we are not remaking cells, but we are able to mitigate the progression. And that is the same when we measured at the 5-month time point. We are also able to control the arrhythmia burden. So this is the arrhythmia burden in a knockout mouse. Those that were injected with the AAV are below the 100 PVCs mark. So it's also having a very powerful antiarrhythmic effect. And this is from the paper of Tenaya. Using our mice, not only do you get more PKP2, and in fact, much more than in control, but you also get a recovery of desmoplakin and a recovery of plakoglobin. So there is a remake of the components of the nuclear envelope. So there is recovery of the desmosomal remodeling, a reversal of the transcriptional reprogramming, and a tolerance to high doses. So in conclusion, AAV-mediated PKP2 delivery arrests the progression of the RV and the LV cardiomyopathy, reduces the arrhythmia burden, did not show you the data on the fibrosis, but it arrests the progression of fibrosis, and drastically improves survival from 100% mortality to 100% survival. Now, I think this quote is very important. It comes from George Box, who said, all models are wrong, but some are useful. Our model is wrong to study arrhythmogenic cardiomyopathy as a clinical entity for all of the reasons that you can think about. But it's useful. It's useful to know the phenotype of a gene that, when mutated, can cause PKP2 arrhythmogenic cardiomyopathy, and then use that knowledge to generate preclinical data that can guide alternatives to therapy. So with that in mind, I think there is a reason for cautious optimism. So these 3 companies obtained FDA authorization. With Rocket, we obtained it in May of 2023. Lexio a few months later, Tenaya a few months later. There was a 4th company, Biomarine, which made public their PKP2 program. We did the preclinical, worked exactly the same way, but decided to get out of the race. And so they did not proceed to IND. And there are other experimental therapies under investigation for helping patients with ACM. So there is reason for cautious optimism, but of course, the gene therapy in humans carries serious risks. It cannot be for everybody, and it cannot be done without a very careful selection of what the ideal patient is. And so with this cautious optimism, I want to just thank you again for your invitation, for your attention. I'm happy to answer any questions.
Thank you, Dr. Delmar. It was excellent talk and beautiful work. Kali?
So you injected the AAV at 7 or 14 days post tamoxifen, I think is what you'd said. But in your model, you showed that you didn't see phenotypic changes in the heart until 21 days, I think it was, when you're seeing collagen infiltration. Have you tried doing AAV injection after you start to see these changes in the heart?
So the very earliest of signs of disease happened at day 14. So that was the latest we decided to go. Because also takes time for this to work, right? For the AAV to infect and then for the transgene to express. So we think that this is more or less where we would want to be with the patients. We don't think that the ideal patient is a patient that has a severe cardiomyopathy, because the heart is gone. At that point, putting more PKP2 is not going to help. You want to be in an earlier stage, and we can talk about who the ideal patient is, and it's very complicated. So it's it's a very narrow window, but that's why we decided to go into the early stage to see that. Steve.
Thank you, Myra, for an inspiring and very thoughtful talk. I've really enjoyed it, particularly the cautionary tale at the very end of it. But now I have a couple of questions. With the AAV dose, can you run them? And do they still maintain 100% survival? If you actually exercise, does that no longer unmask the disease? This is a continuation of our conversation this morning.
I know. And the answer is that we have done the experiment. We have not published it.
Okay.
But it is I have no problem telling you, they tolerate the exercise.
Okay, and then backing up to the very beginning of the presentation. So flecainide, as you know, reduces PVC burden in general. And just using PVC burden as the index of arrhythmia risk is what led to the CAST problems, right? So does flecainide actually prolong the life just as your AAV does?
So in the mice, I don't know. We didn't do that because this was just a sort of a shot in the dark. That's a very good question, but honestly, I doubt it. Because the mice die in biventricular failure. They don't die of arrhythmias. And of course, whether it would prolong the life of a patient would be very difficult because the course of the disease is so variable that you really don't have a timeline like the one that you can get from a mouse model. So that's still to be open. Hopefully will not be the CAST story all over again.
Fascinating talk. I have two questions. First one, do you know by which mechanism the deletion of PKP2 in cardiac myocyte induce fibrosis? Do you know how fibrosis is induced? And the second question, are you going to use this AAV, the one that used in mice, will you use the same in human? I mean, I know that other clinical trials using AAV in the past have been done in the heart, and they didn't show really efficiency in affecting cardiac myocyte. Do you know if this AAV is really affecting cardiac myocyte?
So for the first question, I think that the fibrosis in ARVC is reparative. I don't think there is an invasive behavior of the fibroblasts to attack the heart. A very important component of ARVC that is not recapitulated in mice, not in ours, not in any of the models that there are, is that there is adiposis. There is fibroadiposis. And those are adipocytes. It's not that the myocytes are getting lipid particles. So I think that is reparative, but there may also be other hypotheses. Particularly, there is a hypothesis that we postulated some time ago that the epicardial cells, which are progenitors of fibroblasts, can also differentiate towards the adipocyte line if you don't have good desmosomes in the epicardial cells. That's only a hypothesis. With regards to the AAV, we used AAV RH74. The reason is that's a rhesus monkey AAV, which means that there are less people that are seropositive and therefore not susceptible to the treatment with the AAV. So the AAV RH74 is cardiotropic. The PKP2 is under the control of the alpha TNT promoter. And I'm sorry, yes, the troponin promoter. And So the penetrance in the mice is good. What will happen in humans, it looks as though from other studies, these AAVs are having more or less a 50% penetrance.
Excuse me. Yeah, thanks, Mario. I mean, inspiring and astonishing. It's just such wonderful work. So I'm wondering about the loss of PKP2 and the nuclear ruffling and DNA damage phenotype. And, you know, your question was, you know, how do I link the loss of PKP2 to this phenotype at the nucleus? And in the images of the PKP2, it's clearly only localized at the intercalated disc. So you still have that sort of geographical problem. And so, you know, what mental exercises are you going through?
So the link and the reason why we went after the Desmin paper and studied it carefully is that the desmosome is an adhesion complex that intracellularly anchors to desmin filaments. So, and when you look in the intercalated disc with this type of microscopy, you actually see a separation of the desmin filament from the desmosome in the PKP2 knockout, or whatever is left of the desmosome in the PKP2 knockout. So we figured, naively perhaps, that the loss of anchoring at the intercalated disc was then propagating into a loss of anchoring and architecture across the cell, including then its anchoring and the nucleus. So desmin anchors at the disc, it anchors in the nucleus, and it runs on the Z discs. So we thought you remove the tie-up point for one of these tie points, and you are going to unravel the whole yarn ball until it reflects in what happens in the nuclear envelope. And that is, I think, what is happening. Mechanically, the desmin is not stable enough to maintain its architecture.
Right. So it's kind of like a suspension bridge type hypothesis. Yeah. So, but what about the adhesion itself? Is it, you know, you've you know, that, I mean, that makes sense, but does the adhesion itself also have a role? And, you know, is it the maintenance of that adhesion? Is that part of the phenotype too?
I would love to answer that question. What we do see is that the cells do separate. When you knock out PKP2, you see a separation of the cells from one another. Even in one of the images that I show, you can see that when there is no PKP2, instead of having a nice red line of PKP2, you have a blank because the cells are separating. Now, what is the adhesion itself? What is the contribution to, especially the arrhythmogenic phenotype, is, I think, a question still open for discussion. Is it that you also lose gap junctions? Because you do. Is it that there is remodeling of the sodium channel? Because there seems to be. Or is it all of the above? Because as you know, for arrhythmias, you need two things. You need a trigger and you need a substrate, right? So we believe the trigger is the triggered activity, the calcium dysregulation. But in the substrate, the adhesion component, the loss of gap junctions, the changes in the sodium channel, they may constitute the substrate for the arrhythmias. Now, when in the patients, when the disease progresses, interestingly, they have a lot of arrhythmias. I mean, you saw the arrhythmia burden, but they don't have life-threatening arrhythmias anymore. In exceptions, of course, taken into account. But what you have is just the monomorphic VTs that keep coming because you have the large areas of fibroadiposis. In the beginning is more problematic because you have a lot of mass, you have a lot of volume, and therefore you have more room for fibrillation.
Hi, I really enjoyed your talk. I was wondering, would you argue that arrhythmia risk during the prefibrotic stage of arrhythmogenic cardiomyopathy is more related to microstructural changes such as the nuclear ruffling that you see, or if it's more intercalated disc dysfunction and electrical changes, which kind of relates to the tail end of Rob's question there? Also, it has been suggested by other groups that Brugada syndrome could have microscopic structural changes in the right ventricle that could kind of increase the arrhythmogenic risk because of the substrate differences. Take that with a grain of salt as you will, but do you think, have you seen or looked at other arrhythmogenic models such as Brugada syndrome, long QT, any of the loss of function or gain of function, not in structural specific diseases such as cardiomyopathy that have this nuclear ruffling, or might it could be?
I mean, your questions are excellent, both, and I already forgot the first one. But I will start with the second one. The answer is no, we have not looked at specific models, but it is a very important question. There is a structural reason why we think the nuclear envelope is messed up. You get the same thing in laminopathies. By the way, there is a paper by A.J. Marian last year that showed also a damage of the nuclear envelope in the laminopathies. That doesn't come as a surprise because it's a mutation of the lamin. You get the same in the desmin cardiomyopathies. But it would be very interesting to look at other diseases that are supposed to be pure channelopathies and see what happens, including Brugada. Because I don't think that the sodium channel is there only to pass sodium ions through the membrane. I think that has other functions that are unrelated to electrophysiology, and that may be more of the substrate for Brugada than we think. And with that into mind, there are, as you know, a number of studies showing that there is actually subepicardial fibrosis, particularly in the RVOT of patients with Brugada syndrome. And I am of the belief that that is actually more of a substrate for the arrhythmias than the repolarization abnormalities or the theory of the phase 1 reentry that has been also proposed. Please, sorry, remind me of your first question.
I actually have a different question that I would want to ask
Okay.
instead of my first question. So with the dysfunction in the calcium-induced calcium release that you're talking about, why would we not see PVCs during routine checkups because of the difference in the activity? Why is it still a concealed phenotype, do you think?
So I think that the reason for that is that we in the United States do not include the ECG as a routine phenotype of anyone that is not below 60 years old. So in Italy, and that applies to all inheritable arrhythmia diseases, in Italy, for example, every kid gets an EKG. Not only when they are going to go into varsity sports, every kid gets an EKG. And so the incidence of sudden death in athletes has dropped dramatically in Italy as a result of that. We don't do that here. It has been proposed, it has been fought, and it has been lost. And there are a number of issues that have to do with cost, others that have to do with liabilities. The pediatricians don't want to take the liability. Now, of course, ECGs are being aided by artificial intelligence, so the liability may be less, but still, it's just unknown. So we don't detect them because we don't look for them. But a routine examination of that kid could have shown that, especially under stress, there would be PVCs. And of course, you all may be familiar with these horrible scenes that happen on TV in front of world audiences of athletes, particularly soccer players or basketball players, that fall on the ground. And some of them have been resuscitated. Now the medical teams on the field are trained in advanced CPR, but up until 5-10 years ago they weren't, and they would walk to see the patient with a defibrillator in the hand that they didn't even know how to open. Right?
Thank you very much. It seems like there's a more question. There is a one question online, but we have a reception at 4:30. Please come and please bring a more questions, ask questions at the reception to Dr. Delmar. Thank you very much.
Thank you. Thank you.